Good? Okay. Yay, we're good. We're ready to start. Sorry for the slight delay. We are having technical issues. So welcome everyone to our November Live at Lunch. My name is Alex and I am a learning program developer here at the Royal BC Museum. I just want to say that this Live at Lunch and all of our Live at Lunches usually take place here at the museum on the traditional territories of the Esquimalt and Songhees First Nations, the Kwanghan-speaking people, and we give them thanks for the stewardship of this land. Live at Lunch, for those of you that aren't regulars, is a monthly noon hour lecture series. Usually we have members of the staff from the museum talking or special guests. It lasts about 45 minutes to an hour, and then we have an opportunity for a Q&A afterwards. So today we're very excited to have Andrea Valcourt here. Andrea is a UVic student in her final semester at UVic. She's been working with paleontologist Victoria Arbour since January 2022. And next month, in January, Andrea will be going on to do her master's thesis or master's program at the University of Toronto. So she's going to be talking about what she's been doing with Victoria since January, and she's going to go over some of the projects she's been involved in over the, last year and a bit. Okay, so without further ado, welcome Andrea.
Hello everybody, I'm Andrea. Today I'll be talking about stuff we do in our paleontology prep lab. All right, before I get into that, I'll just talk a little bit about some of the work I've done here before. My very first project was a directed studies, which was looking at these little fish here from the Maccabee fossil beds. This fish in particular is a Eohyodon rosy, and it is one of hundreds of fish we have in the collection, and my job was to catalog them and see if I could tell anything about the environment using them. After that, I decided to do an honors project, and I was focusing on this skull right here, which is a Mericoidodon, which is this decently sized mammal from the Eocene, which looks kind of like a pig. If you want to talk more about that, I'd be happy to answer questions at the end. And now I'm working in the prep lab doing some projects preparing the fossils that we have coming into our collections. I've been working with Calla Scott, our preparator, in the lab doing that for this semester. So to start off with, before we can prep a fossil, we need to find a fossil. We get them two different ways. One is from donations, and the other is from doing field work. For the donations, a lot of times it's just the people in the public letting us know that they found something. There is some laws around it, so you can't dig a fossil. So if you find something really cool, the best thing to do is mark down the coordinates, take photos, and send it to the museum. But if you do find something on the surface that looks like natural processes are going to destroy it, you can pick it up and take it. You still want to mark down the location because that could be important for science, and report it that way. And also, if it's just a small little fossil like the one here, which is a bone fragment, that's not super significant, but if you want to know what it is, you can just ask the museum. So field work requires a permit, and oftentimes is quite remote. So this picture over here is of some of the museum's work at Betsy's Plateau. It is incredibly remote, so they have to helicopter in. And in that particular case, it's more that they know there's bones up there, but they don't know where. So they're scrambling along all these boulders, looking at them, trying to find bones.
Where is Betsy's Plateau?
Northern BC?
It's sort of near Dease Lake, but not really.
So a lot of the fossils that we have to prepare are from these chunks of rock. Other times we aren't exactly like searching for fossils, like this track site where it was reported by a member of the public. We know they're there, and now we're just going there to do work on it. This is me actually taking a quick nap in the sauropod track, which are these giant footprints here. Great for napping. Great back support. So once we have a fossil, we can take it to the museum. A lot of times they're in rock, which we call the matrix, like this, and we don't actually know what it is. So this, we can just tell it's a bone. So we got to take it back to prepare it to take a closer look. So before we can get it to the museum, we don't want it to break during transport, so we have to do some things in order to protect it. So bigger slabs like these, we have to cut down, so we can use this giant, basically a chainsaw but for rocks, to make the rock more manageable. And sometimes we have to use little hand tools like this, where the three of us are using hammers and chisels to chisel out the mudstone layer that's sort of sunk into the track in order to actually see the track in the sandstone. Then we want to clean. Some cases cleaning actually comes first, like this is the spot we were working in the previous picture, but you have to get rid of all that mudstone sitting on top before you can clean it properly. And other times like this, so this is an elasmosaur that was stored at a dam that we have now brought back to the museum. And because it was sitting there for a while, it's got lots of rocks on top. But this is a more special case where we have to clean it really meticulously because some of those rocks are bits of bone. And a cool thing about elasmosaurs is when they were alive, they actually ate rocks, and those rocks end up in their stomach, and that's part of the fossil. But when we're cleaning it, we have to distinguish which rock was the rocks it ate and which rocks are just rocks from the ground. So here we're like sorting through, putting the rocks that go on the ground, and putting anything that we have to take back to the museum into bags to potentially glue it back onto the giant specimen. Once we do that, we glue any loose bits down. The glue is something called B72, which is actually this plastic here, and we dissolve it in acetone, and then it becomes liquid, and we drip it onto the rock, and the acetone evaporates, leaving behind the plastic. And this is perfect because later in the archives, when we decide we don't necessarily want that, you can reuse acetone to get rid of it. So it's not permanent, but it holds things down. After that, we gotta get ready for the protective layer, but we don't want that layer stuck to the rock itself. So we put paper towel, or if we don't have enough toilet paper, and we make this nice layer. We don't soak it, but we have to use water to dampen it to make sure it's actually hugging the fossil. So then we do a plaster layer, and plaster is sort of this powdered rock that we mix with water, and then once it dries, it leaves behind this rock that can make you look like a statue. And then we use either burlap or gypsona bandages, which if you've ever had a cast, that's actually the exact same thing that they make the cast out of. And we soak the bandages or burlap into the plaster water mixture, and then put several layers of that. And at the end, we use any extra plaster to fill in any gaps, and we smooth it out, because if the plaster is, or if the burlap's exposed, all that liquid makes it susceptible to mold, and it could be sitting there in the field jacket for quite a while, and we don't want it to get moldy. Not everything can leave the field. So one example of that is footprints. Footprints are great, but they're often in giant trackways, and you can't exactly take the whole trackway. Plus, it's really important as part of the study to look at how they are in relationship to other tracks. Also, since it's such a huge area, it's important for the current ecosystem. So if you look in the track right here, there's actually a little toad. We named her Irene. She's a female western toad, and because she likes damp environments, water typically pools in these footprints, making it a great environment for her to hang out in during the day when she's sleeping. So we often found her in various footprints every day, and it was really cute. Don't want to take away her home. So one way that we can document these tracks is by mapping. So on the ground, we would make this giant grid, and then each meter by meter, we have this smaller grid that has on the clipboards a matching grid, and someone will have the plumb bob, which is this object that Derek is holding, and they will point to important features on the ground, like the end of the heel or a toe, and the person with the clipboard will make a little dot on where it lands on the grid, and then it becomes like a handmade connect the dots. And then once you have all these grids, you can put them together, and you end up with something like this, where then you can study specific trackways, and you can look at individuals where they're walking and their behavior. But we do want to take closer looks at the footprints, so we make some molds. We use silicone molds because they last a while and they are quite flexible, but it requires mixing a part A and part B, and these are actually two different mixtures of it. The first layer here, which is in orange, is a thinner mixture, and the thinness allows it to get into specific details. And then once that dries, we put on a thicker layer so that it's more solid. And then once that one dries, we put on a third layer, but this time with keys, which are these three little things here, which helps when we make the backing to line up where it goes. That backing is made of plaster, so it's the same sort of thing. We use the burlap or gypsona, and we soak it in the plaster, and then we put it on the back. We smooth it out just so that it doesn't have sharp edges, otherwise we have to grind it down. And then we let that dry too. Once it's dry, the plaster part actually just pops right off. And then because this is stretchy, we have to go around the edges to peel it off. And then once you get a good grip, you can just pull the whole thing. And it's really satisfying, which is, it was so good I had to take a picture of it happening here. Once that's done, you end up with this weird ring around the footprint, which is the plaster, but because it's rock powder, we can easily break off the bits, scrub it down, and then nature washes away the rest. And when you're done, you have your mold, which is ready to be carried down the mountain. And I actually have one of them from this trip here. Is this footprint here. And you can see, so this is the silicone, you can see the three toes here, and then it does pop off the backing. So these are the keys that helps line it up, and then this is the plaster, which keeps it in the shape that it was in the ground. It just pops back in.
What species is that?
I don't actually know, and that's one of the weird things about footprints. Footprints don't often, you can tell from bones how many toes it has, but you can't tell exactly what the foot looks like, which is why footprints actually get their own genus and species names. It's a whole other beast of paleontology. So going back to when we have actual fossils, well, they're all fossils. When we have like bones inside rock, this we can't study, so we have to remove the matrix around it. So we do that by air scribing, which is this object right here, and I have one of them. So this here is the air scribe. This tube connects to an air compressor, and the air is forced through here, which causes this little bit to vibrate. So I'm not actually pushing on the rock, the vibration is chipping away parts of the rock. Because it is breaking off a lot of little tiny bits of rock, you have to use this here, which sucks the smaller bits of rock out of the air and takes it somewhere not in my face. These two objects are incredibly loud, so I have big protective headphones. I also have a second pair of earphones underneath just because it's that loud. With rock chips flying around, I have to wear goggles, even though I wear glasses. I wouldn't want my glasses to get chipped or rocks in my eyes. And I do wear a more heavy-duty mask because bits of rocks don't belong in your lungs. This is an incredibly slow process. So this is what the rock looked like at the start. I actually have it there. After 1 hour of work, you could kind of see I've taken off the top layer, which was quite easy to remove because it was already weathered and already damaged. And then after it was 9 hours of work, it looks quite different. This side, which is here, is quite a lot lower just because it was lots of pebbles, and you can carve around the pebbles, but all the vibration causes the pebbles to just fall apart. But this side is entirely sandstone, and this particular rock is so hard to get through and so well cemented that it barely went down. So it used to stick up above the rest of the rock, and now it's just about level. So this is a time lapse of another student, Emily, where she takes just a small layer of rock off of the top of this rib. And you can see all the marks that the air scribe is making and all the bits of rocks that's coming flying off just from taking off this layer.
It's making me sneeze.
Yeah, so it's a very slow, tedious process. Not all the fossils we get in the museum are in one piece. So this here is a Triassic age fish, which is the sort of starting era of the dinosaurs. It's called Bobacitrania, and it was donated by someone to the museum. And it is broken, but that doesn't necessarily mean someone broke it. It came out of the ground like this. Rocks fault and break and joint. So luckily the person found all the pieces they could and donated them. So the first step to do with this is find out where pieces go. So this here is a drawer of tusks, of mammoth tusks, but it's only the pieces that we know are the outer layer, because the outer layer is a different color. So it's like a giant puzzle, but if someone had removed a bunch of the pieces and possibly thrown in other pieces that don't belong, also in 3D. So after 2 hours of looking at this drawer and testing pieces, I found 2 pieces that go together. And sort of the thought process is, okay, they're similar color. This side and this side look very similar. They do line up, but is a quite straight edge, so that's not necessarily meaning that they go together. But there is also these features of the original tusk that line up, so then they can be glued together. Larger pieces like this you need to make a plan, because if you glue the wrong pieces together first, other pieces then won't be able to fit in. So these are actual photos I did before gluing of me trying to figure out what the best order to glue it is, because with some glues, you only get the one shot. So for this particular specimen, which is here on the table, we needed to use a tougher glue, which was a resin. So you have to mix a part A and part B, but because it cures really fast, you have to do it each time you glue the rock. Once you have these mixed together, you put the glue on both sides of where you're connecting it, and then you shove it together, and you kind of gotta wiggle it a bit just to make sure that all the rocks are, all the like broken edges are lining up where it needs to go. And then you have to hold it there for 2 minutes while it starts to cure. And then you just repeat that until you get the whole thing together. Some other thinner ones like this, you can just use B72, just because it's thinner and doesn't need something as robust holding it together. But once you glue it, you have to let it sit and dry, because if you pick it up, it will fall apart and then you have to restart, and that's really sad. And since some objects like this are three-dimensional, you have to use a sandbag and sort of put it in the shape it needs to be while it sits to dry. So once all of that is done, the fossils then can be put into the collections, but not everything can just sit in a drawer. Some things are too fragile. So some of these is sitting in what we call a foam house, which is where the fish is sitting right now. You first have to get this foam, and this foam isn't like styrofoam, it's more archival, so it lasts longer, and we can take away any like static electricity off of it. And we cut it down to fit into the drawer, and then after that we just place the fossil on top and trace it, and then use an X-Acto knife to cut out the edge. Then you got to make sure it fits, otherwise you have to restart. So you just pop out the trace and line it up with the fossil. And if all fits, then you can put it into the drawer, and you want to leave everything you've done to the fossil with it, just so if future work needs to be done on it, they know what you've already done, because in the past, they were more focused on making it look good, so they'd have a little piece, and then they'd sort of make a plaster thing around it and then just paint over the whole thing so it looked like a large object. Then when people later go to restore it, they have no idea what's been done, and then they find out, oh, it's actually only this tiny little piece, not a really big thing like we thought it was. So now we document that stuff. Larger objects that can't exactly go into the drawers need a more three-dimensional house. So the first step is to protect the fossil, which is what this garbage bag is for, because we don't want to get plaster directly on the fossil. Then you want to make the shape that you need, and you do that using foam, tape, cardboard boxes, sandbags, kind of anything you would do in like an elementary craft. Once you have the shape you want, then you plaster that, but the difference between this plaster and previous plaster is we use fiberglass instead of burlap, because fiberglass is a lot lighter, and we only put 2 layers. But if any of you have ever worked with fiberglass, if it gets in your skin, it is so itchy. So even wearing gloves and full protective equipment, it still gets everywhere. So the smoothing it out process is really important for this. And if sharp parts end up sticking up, you have to fully let it dry in order to sand it down. Once the one side is done, you let it dry, you do the same thing to the other side, let that dry, and it ends up like a clam, and then you're able to put holes in it and put screws in order to keep it fully closed. Right. So fossils are rocks. Rocks tend to weather, so if you have things just sitting around for a long time, you have to do repairs. So that's B72 is great for that because it is reversible should you need to. And the first step is to clean it. This pushes air, so any dust or broken tiny pieces that are too small to glue on get pushed off. And then you drip B72, and the fossil just soaks it up like a sponge. And then the acetone that it's soaked in evaporates, and you're just left with the plastic. This is a bit more tricky because you don't want it to pool, so you use a brush to sort of spread it out. but you need to get the brush wet, and acetone is what you use for nail polish remover, so if you put the brush too far in, it'll take the paint off the brush and you'll end up coloring everything the same color as the brush handle, which is bad. So we also share fossils with other museums who want to study them, but you can't just put them in the mail because sometimes things in the mail break. So you have to make them little cozy beds for their trip. So the first step is you take your fossil and you find a nice sized box for it, and then you cut a piece of foam to fit perfectly in the box. Then you trace the fossil, but this time instead of cutting all the way down, you sort of hollow out a little depression to make it like a nice cozy little bed. And you do that for both sides, so it's like a rock styrofoam sandwich. And I actually would gently shake it just to make sure it stayed in the foam and didn't go flying out. I even double checked it while it was in the box just because you really don't want it in there loose shaking around. And once everything's all snug, then you can put any labels that go with it into the box, close the box, and label the box. If you don't label the box, things can get lost. But that's sad. So we labeled the box. Another way to share things with museums is through 3D scans. So you take your specimen and you take photos going around and around and around. So you have tons of photos. Some of the photos from the track site had over 1,000 photos, so it's a lot of photos. And then you let a computer run, so it's a lot of here sitting, waiting, watching. This is actually a photo of us at the lodge during field work, because you can't exactly, if it doesn't work, you can't just go back and take more photos of the track site, so you gotta make sure it works before you leave. So at night you just sit, watch, wait, make sure everything's lined up, all the photos worked. Once you have this 3D scan, which you can send to other museums, it's super easy to make a 3D print. This is a 3D print of the fossil I was working on for my honors project. It's only the teeth because teeth are really important for mammals, but it's great to have that 3D model because the fossil has to stay here, but I can take that to school and show everybody. So our exhibit has a lot of these finished fossils. This one in particular, you can see the lines from the air scribe. You can see that this fossil, you can see the bone in 3D, whereas before this would have been the part that was exposed that's broken off, creating the cross section. And we have lots of these in the exhibit, so feel free to go look. And I just want to thank Dr. Arbor for supervising, Calla Scott, our preparator, for showing me a bunch of the stuff, Derek Larson for making sure I don't cut off my fingers cutting styrofoam, and for the rest of the BC Paleo team. Thank you. Any questions?